Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about virus. So viruses are acellular non-cytoplasmic infectious agents. So they are acellular. Their structure lacks cytoplasm. They are just a non-living particle outside the host cell. They are smaller than bacteria, so can pass through bacteriological filter. Viruses are transmissible from diseased to healthy organism. All viruses are obligate parasite. Obligate parasite means strictly parasite. They are strictly parasite. They can only live or they can only active inside inside a host organism and can multiply only within the living host cell. Okay. Next, the discovery of viruses. So, it was a collaborative effort by multiple scientists over several decades. So, at first, Dmitry Ivanovsky in 1892 used a filter to show that sap from a diseased tobacco plant remained infectious after being filtered, even though bacteria are retained by the filters. So he used a filter, a bacterial filter, the filter that can filter the bacteria. But after the filtration, he concluded that the remained sap can also infect another plant. So this discovery led to recognition that there were filterable pathogens even smaller than bacteria. Later, Martinus Boyjanink in 1898 called that infectious substance a virus, okay, and concluded that it was a soluble liquid germ or contagium vivum fluidium. He told this as contagium vivum fluidum. Next, Frederick Todt in 1915 discovered bacteriophage, which are viruses that can attack bacteria. Next, Stanley demonstrated that viruses were particle, not fluid. Next, after the discovery of electron microscope, scientists can visualize or observe the complex structure of viruses in details. Okay, so this is the historical background in the virus field. Okay, next. They have many characteristic features, including these are very small, we can't see them in our naked eyes. Next, structure, these are made up of nucleic acid, DNA or RNA wrapped around a protein coat. That protein coat, it is called as capsid. Okay, they protect the genetic material of virus. Genome of virus is either DNA or RNA. Viruses cannot reproduce of their own. They infect a host cell and use host cell's machinery to make the copies of their genetic material. Okay, next, host. Viruses are obligate intracellular parasite. They must live inside a host cell to function. They are host specific. Then activity, viruses are inactive outside the host cell. It is active inside the host cell because they use the host cell's machinery to function. Next, shape, viruses often spiral shaped. They vary in size and shape. Mutation, viruses can undergo mutation. Crystallization, they can be crystallized and remain infectious even in crystalline form. Okay, next, Baltimore classification. So the classification system proposed by Baltimore based on the genome type replication strategy. So according to him, viruses can be grouped into 7 group. At first, double-stranded DNA viruses, those viruses which have double strand DNA as their genetic material. Next group is single stranded DNA virus. Those have single stranded DNA as their genome. Next, double stranded RNA virus. Next, positive sense single stranded RNA virus. Next, negative sense single stranded RNA virus. Next, single stranded virus with DNA intermediate in their life cycle, double stranded RNA virus with RNA intermediate in their life cycle. So, this is the classification system which was proposed by Baltimore, and he regarded these points like nucleic acid, strands, replication sense, mechanism to classify the virus into 7 groups. Okay. Next, what is viroids? One is virus. Viroids means these are virus like. These are only RNA particle, only the genome of virus, but that have the capability to infect. That is called as viroids. This viroids was at first discovered by Theodore Danner. Okay, he discovered that these are acellular particles and virus like only the circular RNA that have the capability to self replicate. Okay, next one is prions. Prions means these are only protein coat, proteinaceous particle without any genome, and that have the capability to cause disease or infect. So that is called as prions, and this was first discovered by Stanley Prusner in 1982. Next, what is lytic and lysogenic cycle? And in this following either of this pathway, virus can complete their life cycle, they replicate. Okay, at first, what is lytic cycle? As the name suggests, lytic cycle comes from lysis. Lysis means breakdown. In lytic cycle, the virus take over the host cell, replicate itself, then breaks the cell, releasing new virus to infect other cell. This cycle is more common than the lysogenic one. Okay, so next is the lysogenic cycle. Virus insert DNA into the host cell where it remains dormant and is replicated along with the host cell DNAs. The virus can become active again under certain conditions and can replicate itself and infect other cells. So, this is the lytic cycle and this is lysogenic cycle. Here, the bacteriophage infect bacteria, insert its own DNA. Then this DNA can cleave the bacterial DNA. Then the virus make their own body, baby viruses inside the host cell, and then they break the host cell and release into the environment. Next, in lysogenic cycle, the viral DNA or genome get inserted or get joined with the bacterial DNA and replicate along with the bacterial DNA. Okay, but sometimes they again become active and again enter into the lytic cycle. So this is the basic difference between lytic cycle and lysogenic cycle. Next is the structure of bacteriophage. So bacteriophage is a DNA virus. Bacteriophage is a DNA virus. Okay, DNA virus that have a head consists of 2000 capsomeres, and these capsomeres are protein in nature. Then inside head, DNA as genetic material is present. Then tail is made up of inner hollow tube surrounded by contractile sheath with 24 annual rings. The distal end of tail has basal plate with tail fibers. These are tail fibers. The tail fibers help the bacteriophage to attach with the bacteria. When they attach with the suppose this is the bacterial cell, this bacteriophage attach with the bacteria with the help of these tail fibers. Then capsid, the cell of phage contains encode capsid protein that protects the genetic material and delivers it into the next host cell. So this is the structure of a DNA virus, that is bacteriophage. Next is tobacco mosaic virus. It is rod shaped with helical structure. Their size is this is 300 nanometer long and 18 nanometer in diameter. Structure, the TMB is made up of 2130 identical protein subunits that forms a helical structure around viral RNA. RNA is single stranded and is located between the protein subunit. Okay, the hollow core of TMB has diameter of 4 nanometer. The hollow core means this. Hollow core is 4 nanometer. These are the capsids. Capsomere, single unit is called capsomere. Inside the capsomere, they have single stranded RNA. Okay, TMB can assemble in vitro, and the process is controlled by pH, ionic strength, and protein concentration. Okay, so this is the structure of tobacco mosaic virus, and it is a RNA virus. Okay, next. So these are very, very important. They have economic importance, like viruses are employed in vaccine preparation, in research, in nanotechnology. It is also used in agriculture to make plant resistant to various stress conditions. It can be used in other industries like cosmetic industry, pharmacology industry, electronics industry. Then this can be used in the preparation of medicines, in research. Okay, so these are various economic importance of virus. Next, use of virus in the production of vaccine. So viruses are used in vaccine production in a number of ways, including it can be live attenuated vaccine. So here we can use a weakened virus. They have the capability to replicate, but it cannot cause disease. Next, inactivated vaccine, use a virus whose genetic material has been destroyed, so it cannot be replicated. Vaccine of hepatitis A. Then vaccine made up of from a part of virus, use of protein or other small piece taken from virus. Example, hepatitis B vaccine, vaccine of shingles, then human papilloma virus. So here the vaccine can be prepared from a part of virus. Next, viral vector based vaccine, use a modified virus to deliver genetic code for an antigen into human cell. This triggers the immune response, similar to what happens during natural infection. So in this way the body can prepare antibody. Okay, then these are some viral diseases. These are some plant diseases caused by the virus. One is tobacco mosaic virus, which is caused by the tobacco mosaic virus in the tobacco plant. Then cauliflower mosaic, then sugarcane mosaic. These are some animal disease caused by virus. These are some human disease caused by virus. So it is all about virus. In a very short time I have discussed everything about virus in very details. So I hope you really enjoyed this session and get benefit from this in your examination. So if you liked it, please hit the like button and please share with your other friends and family members, and please subscribe to my channel Sunanda's Tutorial. Thank you.